In this module, we look at the lessons Mendel learned from his experiment, which we talked about in the previous module. Here's the cross again. I have shown you on the full slide. He noticed that wrinkled seed trait was not expressed in F1 generation. However, it reappeared in the F2 generation. Mendel concluded that the spherical seed trait was dominant on the wrinkled seed trait because the spherical seed trait had masked the effect of the wrinkled seed trait. Mendel worked with his data and mathematically, and he looked at the statistics and concluded that P plant must have 2 units of inheritance for each particular character. And the offsprings got 1 trait from each parent. I will explain that with the help of this diagram also. Additionally, Mendel concluded that during the gamete production, that is the production of pollen or the ovaries or the eggs, one of these pair units is given to the gamete, not both, but just one of them. And resulting zygote contained 2 particles for the same trait. And since the zygote was the product of fusion of 2 gametes. Here on the screen, you can see that if we interpret this data, that by this formula, if we say the spherical seed trait is the capital S, then the parent or P1, the spherical seed plant would be, we can say it had 2 particles. Remember, we don't know that these were the genes at that time. 2 particles of capital S. The wrinkled state is the small s's, so wrinkled seed plant had 2 small s's. When we cross them, as we have said that 1 unit comes from both parents, so the resulting F1 plant would have capital S, small s. So that is why, since the capital S is dominant, that is why all the seeds were spherical and none of them were wrinkled. Of course, today we know that unit of inheritance or the genetic particle is basically gene. Today, that's what we call them. Also, I would like to point out that different forms of the same gene are referred to as an allele. So, capital S and small s in F1 plant are 2 alleles. They are different from each other in this case. They could be similar or different. Also, additionally, since we can see it on our screen that F1 plants have capital S and small s, the phenotype, their appearance is spherical seeds. However, they do contain a gene for the wrinkled seed also, which became apparent in F2 generation, that although the F1 plants had all spherical seeds, however, the F2 generation, the trait for the wrinkled seed again reappeared. It was at the ratio of 1 to 4, but it reappeared. We can also summarize this in a simpler way using a Punnett square, which is displayed on the screen. So we write male gametes on one side of the square, female gametes on the other side of the square, and then we see what type of different combinations we can make. So when we cross capital S, small s with capital S, small s, these traits will separate. That was the objective of Mendel's experiment, to see whether the traits separated or the genetic unit or the particle separated or not. If they separate, then we can have 2 types of gametes from both these plants because they have identical, they have same genotype, capital S, small s. So they can produce 2 types of eggs and sperms. One type will contain the capital S, the other type will contain the small s. So when we make the combinations in this Punnett square, you can see that we can have 2 different types of seeds, one with spherical shape and the other with wrinkled shape. Please also notice that there are 2 seeds which contain one capital S and one small s, 2 of them. Since we have said that spherical trait is dominant, it is masking the effect of the recessive trait, which is the small s, Therefore, our ratio here is 3:1, 3 dominant and 1 recessive. We will continue our discussion of Mendelian genetics in the following module.